this opportunity. I'm very honored to have this opportunity to talk in front of this audience that I believe is made by experts in many different fields. I'm a geologist, field geologist, especially focused on sediments. So what I want to talk about today is mostly geology. I know for someone it might be disappointing because some of you are not strictly interested in geological problems, but at the end of the day, astrobiology has the very challenging task of finding life and tracking the origin of life in many different environments on our planet and beyond. And some of you during your research might have already realized that this task is complicated by the existence of different complex geological processes that are very, very often overlooked when in astrobiological studies. So today, as mentioned before, we're going to talk about early life, or I go as back as the Archean. So today I'm going to talk about Archean life. We know that it's extremely important because it can give us information on the origin and resilience of life. And it has a very strong astrobiological interest because most of the life in the Archean thrived in extreme environments, hydrothermal vents, acidic solutions, very arid or UV bombarded environments. Archean life is also important because it can stretches the limits of the chemical and physical condition that can be withstood by a life form. And most of all, and that will be the topic of today's talk, studying Archean life can teach us how signatures of life can be preserved into the rock record, hence the role of a geologist. So we all know stromatolites, I hope, and these are considered microbialites. There are modern stromatolites, and the presence of these microbialites, they stretch back to the Archean, in fact. So I'm going to talk about some stromatolites, but what I'm going to focus on, and we'll try to be as provocative as possible today, because although we normally consider stromatolites are clear evidence for microbial activity, very seldomly in an context we talk about how these other signatures of life can be altered or sometimes obliterated by processes such as diagenesis and metasomatism. So before I start, I just want to introduce the concept that we need to keep at the back of our mind, which is the concept of taphonomy. Taphonomy is a branch of paleontology that studies the preservations of fossils. Clearly here we are not dealing with dinosaurs or vertebrates, we are talking about bacteria. But even bacteria were alive at some point, like in the Archean, and they all undergo taphonomical processes, such as disarticulation, abrasion, bioerosion. These are clearly all processes that are different depending on the environment and depending on the size of the fossil, but they all lead to the loss of some information in the fossil record. Eventually, part of the taphonomy is the burial. So when the fossils, the living, the remains of the living organisms are buried, and that's when the process that I'm going to talk about today will start, and it's called diagenesis. Diagenesis is defined as the sum of all the chemical and physical processes that affect a rock or a sediment right after deposition. And therefore, even all the biological remains within those sediments are affected. And on top of this, there are other processes that can alter the rock and the fossil terrain, like, for instance, metasomates. So now my punchline is that after all these processes leading from a living cyanobacteria to a rock that underwent diagenesis, we might have a beautiful structure, but at the end of the day, that rock will just be a rock, unless we are able to identify some signatures of the life forms that might have been inside that sediments and probably lost during diagenesis and metasomatism. So clearly, I'm talking about sediments. Well, I'm a sedimentologist, but because they have a very high potential of preserving evidence of life, and including microbial life. But we need to carefully consider what are the preservation potential of these signatures of life. Very often we don't know what is the precursor. So what was the original material that eventually, billions of years later, arrived into the rock record. And in order to understand this, we need to carefully study all the diagenetic processes that occurred in the past 2-point-something billion years. Now, the issue of diagenesis, it's a huge problem for sedimentologists. And it's intrinsic to sedimentological study because it starts with the classification of sediments, especially for people like me that study carbonates. The visual study of carbonates introduce already a sort of bias because when we look at rocks, we need to come up with categories and descriptions and names. And very often, different groups, different scientists describe the same rock using different names and different categories. So when we want to have to use these rocks as proxies for Archean environments or looking for microbial life, we add a further layer of bias because we don't know what kind of sediment was that. We only have the end result of billions and billions of years of geological processes. So today, that was just a very long introduction to present you the idea of this talk. So again, I'm just provoking the audience and trying to emphasize the importance of the job of a sedimentologist in this type of field. So the case study for today's talk is the stromatolitic dolostone of the Transvaal supergroup in Botswana and South Africa. I will start with what for some of you might be a very boring petrographic study. So what we do as geologists when we deal with Archean carbonates. I will add some geochemical data because I want to present you some statistical approaches that can be used in order to reduce the bias in studying these very ancient rocks as much as possible. And finally, I will close with some more data that might help in identifying evidence of life in these Archean rocks. So let's start from the geology. We are in southern Africa, especially in the Taphonomy Craton across southern Botswana and South Africa. In black, you see the extension of the Transvaal basins, plural, and this presentation is going to focus in a very small basin called the Kanye Basin in Botswana. We can We don't need to get into detail in the geological map that you have on the right, but I just want to present you the samples that I'm going to discuss today. They are coming from these two locations in the lower Transvaal supergroup from a unit that is called the Ramonedi Formation. This is just for you in case you want to go and download some papers about it. So in this lower Transvaal, the Transvaal is a very important supergroup in this part of the world. It stretches from the Archean to the Proterozoic, roughly between 2.6 and 2.0 billion years. Very quickly, the basin itself formed around 2.7, 2.8 billion years ago due to crustal thinning. So there was stretching of the crust and clearly formation of some volcanic units, followed by a huge intrusion of granitoid rocks. And finally, once the volcanic activity was stopped, we had some thermal subsidence and the deposition of the first sedimentary unit in the region, which is the Black Reef Formation, around 2.6 billion years ago. This was followed by a major transgression around 2.55 billion year, and that's when the region was covered by a large carbonatic platform, which is what we are going to focus on today. We are in the lower Transvaal in the Taphonomy Dolomite group, and we're going to look at the Ramonedi Formation. That's this huge carbonate platform was covering most of the northern margin of the Taphonomy Craton. So we are between 2.55 and 2.43 billion years. Sorry. So what was the environment in that platform? I got this beautiful image from a website dealing with Archean world, and this could have been pretty much the type of environment that existed at the transition between the Archean and the Proterozoic on the Taphonomy Craton. There might have been no volcanoes, might have been no hydrothermal activity. There might be a lot of things are not clear. And the geological problem, the first geological problem we face is exactly how do we know what kind of environment existed when this carbonate platform was covering this huge part of the continent. The first thing to do would be to have a clear sedimentological understanding of this unit, or what we call in geology a facies analysis. On the left-hand side, you see a sedimentary log of the lower Transvaal supergroup in Botswana, and as I said at the bottom, the bedrock is made by crystalline and volcanic rocks, on top of which we have conglomerates and cross-bedded units typical of transitional facies between continent and sea. We have beautiful wave ripples typical of marine environments, and then finally the entire region is drowned, and you have the first carbonates with stromatolites, dolomitic stromatolites, planar stromatolites, and after around 200 meters, the entire unit is drowned, and we have a chert breccia that indicates another major unconformity in the region. The unit that really matters for this talk, as I said, is the Ramonedi formation, which is mainly made by dolostone, so dolomitic carbonate with large domal stromatolites, as you can see in panel A, stratiform and microcolumnar stromatolites overlain by columnar stromatolites, and the platform sequence is closed by oolitic chert. From a sedimentological point of view, this sequence it's already very interesting because it reflects a drowning of the platform, but we don't have time to dwell into this. The question is if the presence of this stromatolite is enough evidence for life. The question is, of course, because stromatolites are microbialites, but if we look into these stromatolites and we do some microphages analysis, we have very little to be happy with, because most of these rocks are made by microdolomite or microcrystalline dolomite with veins of quartz and plenty of rhombohedral dolomite. And we will see later why this rhombohedral dolomite is quite important. There are very, very few evidences of potential microbial activity like these spherulites that have been found only in one sample. So we do have clear evidence for a process called dolomitization that we'll see in a minute, but very, very little evidences of microbial life. So where are all the microbes that form these stromatolites? The job of the sedimentologist is to classify, as I said, every type of rock that is found. This slide focuses only on the stromatolitic dolostone. I don't want to make it extremely complex for the non-geologist, but we found 6 different textures. So the dolostones is not just homogeneous, it changes, as you can see in these 4 pictures. There are different fabrics or textures in the dolostones, and some of these might indicate the occurrence of a process called dolomitization. So the process of forming dolomite from another carbonate. So this process of dolomitization, it's crucial, but it's even more important when we are dealing with very old rocks, because it might occur at any stage of the geological evolution of a basin. So we have in these slides different models of dolomitization. You might have precipitation of dolomite into a subca environment, into ephemeral lake, in carbon-rich sediments, and also dolomitization due to microbial activity. So the point is to identify what kind of process occurred 2.5 billion years ago. It's extremely different, because all we have so far is some microbial, some textures. This one on the picture is all dolomite. You can see some is microcrystalline, the MDO, and some is coarser. And the 2 different type of dolomite might indicate different type of processes of formation. Now, another layer of complexity comes from a process called metasomatism. This is a real revolution in the mineralogical composition of a carbonate. And it overlaps with diagenesis. In the specific case that we're looking at, you might have injections of quartz and the presence of neomorphic mineral. Neomorphic, it means minerals of new formation, like tog in this case. I was saying that metasomatism, it's a real revolution. Now, I don't want to get into the details of this slide, but what you're looking at, it's these are analyses performed on the stromatolitic dolostones, very close to an intrusion that causes metasomatism. And instead of having dolomite, a lot of different neomorphic minerals appears, like phlogopite, chrysotile, talc. And this means that the system open, the mineralogy change, and everything has been altered. I show you just one example of the rare elements from these samples, where there is a clear enrichment in the light rarer elements, and there is a europium anomaly typical of hydrothermal activity. So the point is that the entire system at some point in time reopened from a geochemical point of view, and everything was reshuffled, reorganized from the mineralogical and the geochemical point of view. So even when we do geochemistry, very often we resort to geochemistry in order to try to sort out all our issues. But even when we deal with geochemistry, we need to be careful in the interpretation of the data. First of all, because often our data sets are very large and noisy. Noisy because sediments have a natural heterogeneity, especially carbonates, and because geochemical proxies are affected by diagenetic overbreed. So in the next few slides, I'm going to quickly show you a statistical approach that can help us to unravel these large data sets and try to bring home some solid information about the rock we are dealing with. And just spoiler alert, you're going to be probably disappointed by the outcomes. So the process is quite straightforward. You first optically analyze your sample. So you do the petrography, like we've seen before, in order to identify the different microphages. Based on the microphages, you do your geochemical analysis, like in this case, some laser ablation in dust-coupled mass spectrometry. And finally, you feed all your database with more than 12,000 entries, in my case, to the statistic, the algorithm, so the artificial intelligence. So in the first step, we asked the algorithm to cluster all our points based on geochemical composition, ignoring my petrographic interpretation. So in a way, avoiding my geological bias. And in this case, the machine learning technique identified 4 clusters based on the geochemistry of on the geochemical data, whereas myself, with the optical microscope, I identified 6. So these 6 clusters, visual cluster, are, for instance, this type of dolostone with very complex margins, rhombohedral dolomite, microcrystalline dolomite, and so on and so forth. So myself and the algorithm were not in agreement. So in the next step, we try to teach the algorithm to think like a geologist and try to identify similarity between the geochemical data set and the optically described microphages. And using a random forest method, we had a 72% agreement with some observation, but the machine learning technique told us that some of the clusters, some of the types that I identified as a geologist were actually clustering together, like these ones that for me were three completely different dolomites, but for the machine learning algorithm, they were all and the same. So the lesson that we learned from this exercise was that, first of all, microcrystalline dolomite, which is the one that might have the most interesting information in terms of microbiology, because you probably know that micrite, it can be often associated with microbial communities. But my identification of microcrystalline dolomite was an oversimplification, because in fact, what was for me just microcrystalline dolomite for the algorithm was subdivided in three different type of dolomite based on the geochemical composition. The second and probably the most important lesson that we learned is that what was that for me, the dolomite had very different crystal shapes and size. And as a geologist, I was taught that the shape and size of the crystal is linked to genetic processes and diagenetic processes. But in fact, for the algorithm, these were identical type of dolomite. So even if the texture were different, they were chemically the same. So that meant that the processes that in our mind were supposed to mobilize elements, so at every step of the diagenesis, were probably not as straightforward as we thought. So this had clearly a bearing on the interpretation of all our geochemical data set. And we used rarer elements because they are often considered as a good proxy for pale environment. And we considered that after the starting of a diagenetic processes, even if the crystals were changing, in fact, their chemical composition was always the same. So there were chances for their chemical fingerprint to still retain some original aspects of the environment of deposition. Now, that means that if diagenesis didn't overprint completely the rarer element distribution, we might give a try to stable isotopes, especially carbon and oxygen. You know, they are very often used for environmental reconstruction. So we did some test analysis. These are not published, but hopefully they will be soon published. But to my greatest disappointment, although rarer elements were giving us some hope, stable isotopes were clearly recording diagenetic processes with temperature up to 180 Celsius degrees, so quite deep burial diagenesis. So what is left for us to do? Because the situation is quite dim right now. We try to look for other potential evidence of biogenic activity, for instance, the presence of pyrite. And we found some pyrite grains. Unfortunately, most of these pyrite grains were heuhedral, and they had a shape and a sulfur composition that points toward most likely a volcanic origin. And the only, and it was really only one, pyrite grains with some sort of rhomboidal shape that might have a link with microbial activity, was too small and didn't yield any sulfur, usable sulfur data. So once again, we are back to square one, where we cannot have unambiguous evidence of the activity of microbial community. Now, as we probably everyone is expecting this, probably our last resort is going to be Raman spectroscopy, because if there is any organic matter or kerogen left, Raman spectroscopy might be the right tool. I know there are many other techniques, but I cannot cover all of them right here. I'm telling you what we are busy doing. I don't have results yet, so I'm just presenting some other teams' work. So Raman might be our last resort, really, to check for the presence of some organic matter and eventually have a direct link with microbial community. And that is even more important, considering that we know there is a Raman on Mars as part of Sherlock on the Perseverance rover, although it's probably malfunctioning right now, but anyway, it has a strong link with the exploration of Mars. But that is still an ongoing part of our work. So in conclusion, yes, Archean stromatolites, they are very interesting from an astrobiological point of view, because they are very, very old, and because they are extreme. Well, this is just to say that they probably form into an extreme environment. So these both are qualities for a good analog, especially if we look at Mars. But unfortunately, many of the Archean stromatolites that we have, and not just the Transvaal one, they have very few micromorphologies that can indicate microbial life. So there are no fossils. And the reason is that this dolomite is recrystallized, sometimes in multiple stages due to dolomitization and diagenesis. So the fossils that were replaced by minerals, they've been obliterated by the neomorphic processes of these crystals that change their shape. So even though we have good evidences for diagenetic processes not affecting the chemistry of the dolostone, at the very end, we had some metasomatic processes that, on the other end, completely changed the chemistry of the rock. And we are not yet sure of to what extent the geochemistry was modified, but we know it happened because we have a completely new set of minerals. So microcrystalline dolomite is our last chance to have some pristine, some evidence. Evidences of to have some evidence of microbial activity, because they might be considered as pristine products of marine diagenesis or deposition. And inside this microcrystalline dolomite, rarer elements might still be used with all the due consideration of diagenetic effects. Some important proxy for the reconstruction of paleoenvironments, but they won't prove the presence of microbial activity. They will simply pinpoint an environment that might be suitable for the development of microbial communities. So, as I said, this might be a bit disappointing. Consider that is not. I didn't present all the existing techniques that can be used. This is what we've done. This is what I know, and some of you might have different experience. But the bottom line is that diagenesis, it's very often overlooked, and yet it has a very important bearing on any interpretation that we do of Archean carbonates. With this, I thank you for being so patient to listen to this very long geological talk. And if you have time, I invite you to go and visit our project website, especially Fast for Future, because we recently launched a Center of Excellence, which is very similar to the place where we are today, is a virtual community focused on Africa space science. Thank you very much.